Hello. So today's topic is cell biology, and the question which I have chosen is from GATE 2021 Excel Geology part, and it is a question of 2 marks. So question number 15, it asks us to match the cell organelles in column 1 with the appropriate functions in column 2. So in column 1 we have the cell organelles which are peroxisome, endoplasmic reticulum, glyoxysome, Golgi complex and centrioles. In column 2 we have the appropriate functions: conversion of lipid to carbohydrate, oxidation of fatty acid, N-linked glycosylation, microtubule organization center and O-linked glycosylation. So, to answer this question, we first have to know the various functions of these organs, which we will see one by one. So, first of all, we have the peroxisome. So, peroxisomes are present in both animals as well as plants. And in animals or humans, they have some functions which are beta oxidation or degradation of fatty acids, that is, they can metabolize the fatty acids. It contains catalase, so it acts as a free radical scavenger which helps in the reduction of hydrogen peroxide. So many a times during stresses, there is an increase in the level of H2O2 or hydrogen peroxide radicals inside the cell. So peroxisome, which contains the catalase, it helps in the reduction of hydrogen peroxide and reduces the inflammatory or the reactive oxygen species generation inside the cell. Peroxisomes also have enzymes which are involved in the synthesis of cholesterol, which some of the enzymes are HMG-CoA. Then it is also involved in the synthesis of bile in liver. It helps in the synthesis of lipids to produce the myelin. For example, the myelin sheet that covers the axons or of the nerves, and it is also involved in the detoxification of alcohol from the liver as well as breakdown of excess purines. So these are some of the functions in the animals. Uh, so there are some other special functions in the plants such as the glyoxylate cycle. It has the enzymes to perform the glyoxylate cycle. It also helps in photorespiration, that is respiration that takes place in conjunction with photosynthesis, and it also helps in the degradation of purine. So this is the basic structure of peroxisome. It is present inside the cytosol. It has a lipid bilayer and sometimes it has a crystalline core, and inside the lipid bilayer there are enzymes which helps it to perform all these functions. Next, we move on to endoplasmic reticulum. So, endoplasmic reticulum basically it is it provides the structural framework for the cell, and these are some of the functions. It helps in protein processing and secretion. It acts as a supporting framework for the cell. It helps in the distribution of protein from to and from the various parts of the cell. It helps in carbohydrate metabolism. It also transports modified lipids and carbohydrates to the cell membrane. It has sites for aerobic respiration. It sometimes it is covered by ribosomes, and then it acts as site of protein synthesis. Like you can see in this picture as well as in this picture, this is a TEM image of the endoplasmic reticulum. You can see these small dotted structures on the endoplasmic reticulum. These dots are the ribosome, and when these dots are present on the ribosome, they are present on the endoplasmic reticulum, then uh, so the ribosomes do the protein synthesis, and the protein is directly sent into the endoplasmic reticulum for other kinds of modifications. Then when there are no ribosomes attached on the endoplasmic reticulum, then it is called a smooth endoplasmic reticulum, and it helps in the synthesis of phospholipids as well as steroids. So the RER, since the protein directly goes into the RER, it helps in the folding as well as the modification of proteins One of the modification of proteins is the O-linked glycosylation. So what is O-linked glycosylation? We'll know about it in the upcoming slides. So, endoplasmic reticulum also helps in detoxification of drugs as well as it acts as a reservoir for calcium ions in the sarcoplasmic reticulum, which is present in the cardiac as well as skeletal muscles. So, sarcoplasmic reticulum is the special kind of endoplasmic reticulum which is present in the cardiac muscle as well as the skeletal muscles, and it acts as a warehouse for calcium ions which are released when contraction of the muscle takes place. Next, we move on to glyoxysome. So, glyoxysome is a special peroxisome present in the plant cell, and it is a fat storage tissue of the germinating seeds. So, it possesses enzymes of the glyoxylate cycle. It contains enzymes to break down the fatty acid. So, and it synthesizes sugars by gluconeogenesis. Seedling then uses this sugar to convert it sugars synthesized from fats. Okay, they break down the fats to synthesize sugars, and the seedling uses these sugar molecules until it starts its photosynthesis. It also participates in photorespiration and nitrogen metabolism in the root nodules. So, what are the characteristics? Is that it is a single membrane and the matrix is granular due to presence of various flat globules, and it is only present in the plant cells. Next, we have the Golgi complex, which is present both in the plants and animals. Secretion is the major function of the Golgi apparatus. Then, the modification and packaging of various materials in the vesicle. So, Golgi is composed of tubules and vesicles. So, the modification of and packaging of various materials happens in the vesicles. These vesicles can then fuse with various different organelles like the cell membranes or the different organelles like lysosomes, and then they can transport various proteins or enzymes. Secretion of proteins, secretion and synthesis of glycoproteins happens in the Golgi complex. Formation of cell wall, cell plate, new plasma lamina during the cell division. It also helps in the formation of the primary lysosome, which contains enzymes for the degradation or the apoptosis, as well as degradation of foreign substances, as well, and it also helps in the formation of sperm acrosome. So, sperm acrosome contains the Golgi bodies, which which secrete hydrolytic enzymes, which are essential for degradation of the extracellular matrix of the ovum, so that sperm can reach the ovum. It also helps in the recycling of membrane, facilitates glycosylation, that is addition of carbohydrates to the proteins, liposylation, addition of lipids to protein or to formation of lipoprotein, sulfation, addition of sulfates, and phosphorylation, addition of phosphates. So it helps in various kinds of modifications of the protein in the form of glycosylation, liposylation, sulfation, and phosphorylation. So, as we are talking about the modification of the protein, so there are various kinds of post-translation modifications on the protein. The major or the most important ones are phosphorylation, which happens on serine, threonine, or tyrosine amino acids on the protein because they contain an hydroxyl group and they function in cell signal. Then we have methylation, which is the addition of CH3 group or the methyl group, and which happens on the lysine or arginine amino acid on the protein. They help in transcription regulation as well as protein-protein interaction. Then we have acetylation, which can happen on glycine, alanine, serine, methionine, and aspartate. They promote membrane association. Ubiquitination, addition of ubiquitin molecules on proteins, happens on the amino acid lysine, and it helps in the signaling of proteins. Lastly, we have glycosylation, which can be of two kinds, N-linked and O-linked. N-linked happens on uh, the sequence is given mainly on amino acid arginine, and it has the sequence N, X, and S. X can be any amino acid, and it has to be followed by S or T, or that is serine and threonine. And O-linked glycosylation happens on serine and threonine, and these help in the protein function, protein folding as well as functionality. It gives various different kinds of function to the protein. So as we'll go into a little bit detail about N-linked glycosylation. It happens on the inside the endoplasmic reticulum. So this is the process. The protein goes inside the endoplasmic reticulum, and after that there are various additions of carbohydrate molecules on the protein, and it happens on the asparagine residue on the protein inside the endoplasmic reticulum. Therefore, it is called, as it happens on the asparagine residue, therefore it is called N-linked glycosylation. And we have the O-linked glycosylation. This happens in Golgi bodies, and it happens on the serine or threonine residues of the amino acid. They are essential for the biosynthesis of mucin, critical for formation of proteoglycan core proteins that are used to synthesize the extracellular matrix components. Antibodies are heavily O-glycosylated. It occurs on the serine and threonine side chain of the Golgi apparatus. So this is about O-linked glycosylation. So we come back to the question. So here we can answer the question. So peroxisome. Now we know that peroxisome helps in the oxidation of fatty acids. So P, for option P, the correct option is 2. So we have A and D, which have P as the correct option is 2. So we can have these two options as right, and we neglect the other two options. Then we have endoplasmic reticulum. We heard that endoplasmic reticulum N-linked glycosylation takes place in endoplasmic reticulum. So option number Q, the right option is 3. So here both of these options A and D contain Q as 3. So we move on to the next question, next option, which is glyoxysome. So glyoxysome is a special kind of peroxisome, and it helps in the conversion of lipid to carbohydrate. We saw there that it helps in the conversion of lipid to produce the glucose molecules, which helps the seedlings to survive before they start photosynthesis. So in glyoxysome, the correct option will be 1. So here R is 1 and here R is 5. So this is the wrong option and this is the right option. So S also we'll see. S, we know that O-linked glycosylation happens in Golgi apparatus. So this is the right option. And for S is 5. And lastly we have the centrioles. Centrioles help in microtubule organization. They are present in animal cells or human cells, not present in the plant cells, and they help in microtubule organization during the cell division. Here we have it. The centrosome, this is the structure of the centrosome. They have the 9 triplet tubules. This is how it looks from the top. They have the 9 tubules, and they have central, or they have 2 pairs of central part. They have 2 pairs. So they have the 9 plus 2 arrangement. They form the basal bodies which give rise to axonemes of cilia and flagella. They are always present in pair. They are the component of the centrosome, and they self-replicate during the S phase of the cell cycle, and they help in the microtubule organization. So we have matched all the right options, and the correct option is